Hello, I'm Jack.
And I'm Loudon. Today we're going to talk about hyperspectral imaging and how it can be used to evaluate plant health.
So what is hyperspectral imaging?
To start, let's talk about the human eye. You see color using what are called cones. There are three types of cones which detect red, green, and blue light. Your brain then processes these three colors and creates a composite image containing all the colors you can see.
So hyperspectral imaging works in a similar way. It will scan a wavelength filter over a detector to obtain the amount of light reflected from a scene. If we repeat this with around 150 filters, the result will be a data cube containing information from all the wavelength filters. Here's a demonstration of this, where the camera represents the detector with the filter moving across it. Now this, in and of itself, is fairly interesting, but what can it be used for?
In fact, hyperspectral imaging can be used to evaluate plant health. A healthy plant, such as the one shown in this diagram, will absorb red light at around 650 nanometers as it is used by chlorophyll in photosynthesis. However, it will reflect longer wavelengths in the near-infrared spectrum at around 850 nanometers because the chlorophyll will not absorb it. In this diagram, the black ray, representing the near-infrared spectrum, is reflected off the plant heavily. However, the red ray, representing the red light, will be heavily absorbed by the plant.
Stressed plants will reflect more red light, often giving them a brown appearance. Additionally, they will absorb more near-infrared light due to the breakdown of the chlorophyll cells. Here you can see that the red light is reflected at a higher rate in the stressed plants than the healthy plants. Additionally, the near-infrared light is not reflected nearly as much in the stressed plants as the healthy. It is thought that plants develop this mechanism so that they do not overheat from unnecessary light absorption in the NIR range they cannot use for photosynthesis.
In order to evaluate plant health, we use the Normalized Difference Vegetation Index, or NDVI, which relates the total amount of light reflected in the near-infrared range to the amount of light reflected in the red range.
As you can see from this equation, a healthy plant will have a value closer to 1, while an unhealthy plant will have a value closer to 0. If we compute this equation for every pixel in a hyperspectral image, we'll be able to see overall plant health across the image.
For our setup, we used a uniform white light source, a SnapScan VNIR hyperspectral camera made by IMEC, and an adjustable aperture and 3 plants. The light source was very important because we needed it to be close to a uniform emission across the visible and NIR range so that we could utilize the NDVI equation. It was fairly difficult to arrange the camera system as we wanted the maximum amount of plant in the photos, while also keeping the image sharp. We arranged the plants, then removed them to take a flat field photo. This allowed us to calibrate the camera to the light source, accounting for any differences in the source and detector. After calibration, we imaged the plants using a program called HSI Studio. This takes a few seconds as the filter has to move across the detector.
After obtaining our data, we used MATLAB to evaluate it. Here are some of our results. You can see very clearly the healthy leaves versus the unhealthy leaves. Even at a glance, the NDVI data is easy to understand and evaluate where unhealthy leaves are located. Additionally, you can tell the differences between leaves that were upright versus upside down, as shown in this image, where the rightmost leaf was upside down. We also used HSI Studio to create these images, where areas of similar wavelength characteristics are highlighted. This allowed us to gain a better understanding of how certain parts of the plant reflect light. Overall, our experiment verified that hyperspectral imaging is an effective way to determine a plant's health.
You may be asking yourself, how does this application of hyperspectral imaging apply to the real world? If we were able to increase the FOV and focal length of this camera and implement it on a drone, one would quickly be able to evaluate the general health of a farmland or forest and identify areas which might require more attention.
This specific camera is currently being used at the University of Arizona to identify esophageal cancer for 3 groups of people: non-Hispanic whites, people older than 50, or people with a smoking history. Because esophageal tissue scatters light different in the 470 to 900 nanometer wavelength range compared to regular tissue, this camera can easily identify the affected areas.